Hi, everyone. Welcome to the ARO seminar series. This seminar will be entitled Microglial Roles in Auditory Brainstem Development. My name is Mark Warshall. I will be your moderator for today's webinar. I'm a professor of otolaryngology at Washington University in St. Louis, and I conduct research on how inflammatory cells influence inner ear development and repair. Today's speaker, Karina Cramer, is a longtime colleague, and I'm very excited to hear her talk. First, I'll give you some background information on Dr. Cramer. She's currently a professor at the University of California, Irvine, in the Department of Neurobiology and Behavior. She obtained her PhD from Caltech, where she studied neuromuscular development, then went on to pursue postdoctoral training at MIT on visual system development. And then this was followed by additional postdoctoral studies in Ed Rubel's lab at the University of Washington, where she studied auditory system development. Since establishing her own lab at UC Irvine, she's investigated developmental mechanisms that underlie the assembly of auditory brainstem circuitry, particularly in regions that enable localization of sound sources. Her early research demonstrated roles for the F and efferent family proteins and axon targeting and formation of tonotopy in the auditory system. And she and her colleagues have identified some new axon guidance roles for molecules in the apoptosis pathway. Her ongoing research also explores the functions of glial cells, including astrocytes, microglia, and oligodendrocytes in the maturation of auditory brainstem circuitry. So before getting started, I just want to mention and review a few of the meeting tools that you as an attendee can use throughout this presentation. I'm sure everyone's familiar with Zoom. These tools are located at the bottom of your screen. So first, to keep questions from becoming distracting to our presenter, if you would like to ask questions, please utilize the chat function and direct those questions to me, the moderator. And you're welcome to submit questions at any time throughout the presentation by typing them into the chat function and clicking the send button. Any questions that we don't get to or are not addressed during this session will be answered by the presenter in a follow-up email. Also, closed captioning for this meeting is available for your use, and this session is also being recorded and will be made available for additional viewing. And we hope that you will also participate in our post-webinar evaluation so that we can gather feedback on this presentation. And finally, if you're experiencing any technical difficulties with this presentation, please email the ARO office, and the email address is really simple. It's headquarters@aro.org. That's headquarters@aro.org for assistance, and hopefully we can resolve the problem. So without any further delay, I'd like to turn it over to Dr. Karina Cramer.
Well, thank you very much, Mark, for the nice introduction, and thanks to the organizing committee for in organizing these seminars and inviting me to participate. So today I'm going to tell you about our research on the roles of microglia in auditory system development. Okay, so my outline today is I'll go over a brief summary of the auditory brainstem circuits that we're studying and give you a sense of what kinds of challenges the auditory system faces during development. We'll look at some of the glial cells involved in auditory circuits and consider how they might contribute to building those circuits. And then we'll focus on microglia, specifically, I'll tell you about some of the studies that we've done to look at what happens when you eliminate microglia during development, when you allow them to come back, and what happens when you delete genes within microglia. Okay, so getting into the auditory circuits, this is a very schematic summary of just some of the auditory pathways that we're looking at. So the auditory sensory epithelium maps frequency, but doesn't map space. And as a consequence, the brainstem has adapted several pathways to allow us to infer the locations of sound sources. And in this pathway shown here, this is a pathway used for high frequency sound localization. The input from the cochlea comes here to the ventral cochlear nucleus. The shading shows the tonotopic organization. So we have neurons in VCN that project across the midline and synapse onto neurons in MNTB. MNTB in turn provides inhibitory input to neurons in LSO. At the same time, the neurons from the contralateral VCN send excitatory projections to LSO, so that each LSO neuron receives a tonotopically matched input from both VCNs. And it uses the balance of excitation and inhibition to determine sound source localizations. So we're interested in the processes that give rise to this circuit. And in particular, we're looking at the formation of these specialized synapses and at synaptic pruning. So some of the specialized synapses that we see in this circuit include the end bulb of Held. This is the very large synapse that comes from the spiral ganglion neurons and terminates on neurons of the ventral cochlear nucleus. And we'll look at a lot of pictures here of what's called the calyx of Held. This is a large one-to-one synapse that terminates on MNTB principal neurons. Okay, so we're interested in how glial cells regulate the maturation of these synapses and the formation of these specialized synapses and synaptic pruning. And these are some of the specializations that we see here. And in addition, there are specializations that regulate the timing of inputs as well, so that LSO, for example, gets inputs from this VCN and this VCN at the same time. Okay, so what are the glial cells and how do they contribute to circuits? So this is the calyx of Held, and we've been talking about the principal neurons of MNTB and their calyceal inputs from VCN. But as we can see here, there are many other components to the synapse. In particular, we have these astrocytes that are present throughout the synapse, and we have these microglia shown in green, and we're going to be focusing on the roles of these microglia. So microglia are the immune cells of the central nervous system, and they populate the brain and the auditory brainstem in early postnatal development. But they enter the brain during an very early embryonic development. So we know that early markers for astrocytes and microglia, that is markers for early astrocytes, are seen in very close apposition to labeled calyces of Held at postnatal day 6. The calyces are shown in red and the markers are shown in green. And then if you look a little bit later at P14, we have markers for mature astrocytes, such as GFAP, which are seen around the calyx and much intercalated into the calyceal ramifications. So what about the microglia in the auditory brainstem? So we know that there are microglia in the brainstem at postnatal day 0. Not very many of them are here in the MNTB, as shown with marker IBA1. By postnatal day 8, there are lots of microglia. And by postnatal day 14, we can see the microglia peak in number, and they also have this highly ramified, thick appearance. They taper off a little bit after that and then maintain moderate levels of labeling in subsequent ages. So we first got interested in the roles of microglia because of the timing of the peak in microglia. We know, looking at this timeline, if we look in MNTB, there is calyx formation at these early stages, and we have synaptic pruning, which goes on beginning at about postnatal day 3. And then we have calyx growth that takes place over a long period of time. Okay, so another thing to consider is that this peak microglial density, which occurs in this window here, is really kind of associated with the onset of hearing. So a little bit about microglia that's really interesting is when we think about astrocytes and oligodendrocytes, which are considered macroglia, those derive from the lineages that make the same lineages as neurons. Those are the neural cell lineages, and they are part of the neural tube. But the microglia derive from the hematopoietic cells, and they're in a lineage that branches off early, whereas later cells give rise to things like macrophages and monocytes, which are also immune cells that have important roles in development. Microglia have important roles throughout the lifespan, and as I mentioned, they populate the brain very early in development, and they're important for apoptosis of neurons and also for pruning of synapses. And I've indicated in here some of the signaling molecules that are used by microglia to communicate with neurons, and many of these have been shown to be important in processes of synapse maturation and pruning. Some beautiful studies from George Spiro's lab have demonstrated the process of synaptic pruning using EM tomography in the developing brain. And what they saw is that there are numerous small inputs early in development. Later, there are larger inputs and just a few of them. And then there's a dominant input that is seen covering most of MNTB by postnatal day 6. So when we look at this, we can see at early stages, there are numerous inputs that are called sort of protocalyces. Then there are fewer inputs, a dominant input, and then there's monoinnervation, so that a single calyx innervates each MNTB cell. So to begin to understand how microglia contribute to maturation, we designed some experiments to eliminate microglia during development. So to do this, we found a way to eliminate them. We then wanted to test whether depletion of microglia resulted in changes in astrocyte populations, to test whether microglia depletion changes synaptic protein levels within the MNTB and elsewhere in the brain. And then we wanted to test whether microglia are important for the pruning of the excess inputs to MNTB neurons. So to do this, we collaborated with Kim Green, who's here at UCI, who showed that if you inhibit the CSF1R receptor on microglia, you can eliminate all of the microglia in the brain. And the method that he used was used in adults, and it was fed to them as part of their chow, and they would have sustained levels of the inhibitor. So we actually couldn't use that because we began these experiments before the mice were weaned. And we found another CSF1R inhibitor, BLZ945, that could be given in a sustained level through subcutaneous injections beginning at postnatal day 2 and 4 and 6. One group was perfused at P8, another group was continued until P13. And these studies show that if you look at microglia using IVA1, the BLZ completely eliminates microglia at both ages. This is the aerial expression of IVA1 within MNTV, and this is the actual counts of microglia. And by any measure, there are no microglia after the administration of this drug. We then looked at the effects of microglia depletion on astrocyte markers. We looked at 2 markers of astrocytes that are expressed very early in development, ALDH1L1 and S100 beta. And we saw that there were no changes in the levels of these astrocyte markers. However, when we looked at GFAP, which is really only expressed in sort of late embryonic postnatal development, there was a decrease in the levels of GFAP expression in the animals treated with BLZ 945. We also showed that these aren't a separate population of astrocytes. These are actually the same population. They co-express some of the other markers in later development. And this suggests that the treatment with BLZ 945, which eliminates microglia, results in a decrease in maturation of astrocytes. We looked at expression of VGLUT1 and 2, which is a presynaptic excitatory marker. And we looked at the synaptophysin, another presynaptic marker. And we looked at expression in MNTV at both ages, and we saw no effect of microglia depletion. Similarly, we saw no effect of the inhibitory presynaptic marker, VGAT, and we saw that the treated and control animals had similar levels at both ages. We then went on to look at the pruning of excess synapses in MNTV. So the strategy that we used to do this is we made small injections of rhodamine at the midline and delivered the dye through electroporation. And this resulted in sparse labeling of calyces on both sides of the brain. We then took these sections and counterstained them with VGLUT immunofluorescence. So this resulted in MNTB with some cells having red calyces filled with rhodamine dextranamine or RDA, and all of the presynaptic inputs labeled with VGLUT immunofluorescence. So then we looked at all of the cells that had a red calyx, and we asked whether they had additional inputs that were labeled in green aside from the RDA labeled calyx. And those were considered polyinnervated MNTB neurons without the VGLUT outside of the RDA labeled calyx. Those were considered monoinnervated. And as expected, we found monoinnervated neurons in MNTB at both ages. These are P8 and P13. So you can see this is an example of a monoinnervated neuron here. You can see very little VGLUT labeling. Here, the VGLUT labeling and the RDA labeling are interspersed. And at the same time, we saw polyinnervated neurons at both ages, and you can see them here at P8 and P13. However, when we treated the animals with BLZ 945, we saw that there was significantly more polyinnervation in the treated animals at P13. We didn't see a difference at the early age. So if we look at polyinnervation here, you can see there's lots of polyinnervation at postnatal day 8. And in control animals, there's very little polyinnervation. But with BLZ, there's quite a bit, with about 40% of the neurons we examined showing polyinnervation. So this was a little bit counter to some of the literature on this time course of monoinnervation. And so we ran another set of studies to just make sure that we weren't mistaken. And what we did here is, rather than label the inputs in two different ways, we double labeled with sparse labeling of RDA and a green dial, Alexa 488, to label as many calyces as we could. And in every case, this is untreated animals and control animals at P8 and P13. And in every case, we found examples of cells with red and green calyces, which really indicated that the normal pattern of monoinnervation might be slower and more protracted than previously thought. So since we found an effect of microglia depletion on polyinnervation, we wondered whether microglia are actually eating or phagocytosing components of the excess calyces. And to begin looking at this, we labeled presynaptic proteins, V-glute, and we did this in a line of mice that expresses GFP within microglia. So when we looked at this, we also did deconvolution confocal microscopy and looked at co-localization of V-glute and GFP. We also used a marker, CD68, which labels lysosomes. And if microglia are actually phagocytosing components of presynaptic terminals, then they would be preferentially in these lysosomes. And we found that that was, in fact, the case. There was a great deal of co-localization of V-glute within these lysosomal volumes. And this is consistent with the direct role for microglia in pruning these excess calyces. So these experiments show that treating animals with BLZ 945 early in postnatal development, first of all, eliminates microglia from the auditory brainstem. And we showed that this results in impaired synaptic pruning, which is evident at the later age that we examined. We also showed that this is likely due to presynaptic proteins being engulfed by microglia during development, and presynaptic components may be fully engulfed by microglia. So we also showed earlier on that that there was a delay in astrocyte maturation, or an impairment of astrocyte maturation, and there's a possibility that this could be also part of the problem with pruning, because you might have, if you have fewer mature astrocytes, and astrocytes also contribute to pruning, then they wouldn't be able to carry out their role. Okay, so what happens when microglia come back? So our colleague, Kim Green, showed that when you eliminate microglia with a CSF1R inhibitor, and then you withdraw the treatment and restore the animals to their normal diet, after a few days or a week, the microglia start to come back. So we wondered whether microglia can come back after we eliminate them in the early postnatal period. So what we did was we continued our treatment as we did for the P13 endpoint, but instead we allowed the animals to survive. We perfused at P21, 28, and 7 weeks. So when we did this, we looked at microglia using IVA1 in control and BLZ 945 treated animals. So these are the control animals. Here we have AVCN, LSO, and MNTV. And when we treat the animals with the drug, we see that at P14, there are no microglia. At P18, there were microglia in AVCN, just a few. At P21, they started to appear within the LSO region, but not yet in MNTB. And then by P28, all three of these nuclei were at normal levels of microglia population. And one interesting thing about this is that when we see microglia appear in normal development, they enter the brainstem from this same lateral to medial movement that we see during the repopulation with microglia. So just showing you with longer time periods, we see that you have IBA1 labeling retained up through these late time points in both the control and BLZ treated. And this is just showing that, that we have the same levels as controls by these time points. Okay, we next looked at what happens with GFAP, and we see that GFAP remains slightly reduced at this 4-week period, and that's the same as what we saw with P13. But by 7 weeks after microglia repopulation, GFAP levels reached control levels. And then we also looked at pruning, and we found that after we let the microglia return, MNTB neurons are restored to their mono-innervated state. So this is the polyinnervation here. We saw an increase in polyinnervation at 3 weeks, and at 4 weeks, there was no significant difference. So this implies that the returning microglia can somehow help the auditory system restore its normal developmental sequence. Okay, so because the animals were now 4 weeks old or older, we were able to measure auditory function, and we did ABR recordings in these animals. So one of the things that we noticed immediately was that the treatment of with BLZ led to increased thresholds, and this was in response to both clicks and low frequency tones. And here you can see some of the traces that we obtained. Another thing that we noticed is that when we looked at interpeak latencies, particularly between peaks 2 and 3 and 1 and 3, we found that the latencies were elevated significantly in the BLZ treated animals compared to controls, and that some of these increases in latency went away by 7 weeks. So that was an interesting observation that we made, suggesting again that withdrawal of this drug and return of microglia can help the auditory system return to its normal function. So with these studies, we showed that when we allow microglia to return by withdrawal of the BLZ treatment, we get repopulation of microglia in a manner that sort of recapitulates the normal developmental sequence. We get recovery of pruning and normal levels of GFAP in the auditory brainstem. The treatment and removal of microglia results in elevated thresholds and increased latencies. The latencies are mostly recovered by 7 weeks, which is about 5 weeks after the return of microglia. So we then wondered, well, what if we didn't let the microglia come back? I mean, maybe the changes in the circuit would restore anyway. But what we did here was we developed a paradigm for long-term depletion of microglia in these animals. So we did some pilot studies with the subcutaneous BLZ treatment, and we showed that after a while, it just wasn't strong enough or sustained enough or a high enough dose to work for the long term. And we also knew from our previous study that the rodent diet with PLX 5622, which is a drug that our colleague used in older animals, we were unable to get this drug to work through lactation or through the pregnant moms. And so what we did is a combined regimen where we gave the animals VLZ 945 and at the same time put the moms on the PLX rodent diet. And so once the animals were weaned, then they too were on the PLX. So they, using this regimen, we show that the mice really never had microglia throughout this period. So we then wondered, well, how does that affect auditory system development? So when we did this, we found that there were far worse changes in the ABR. So there were decreased amplitudes, increased thresholds, and longer latencies, and longer interpeak latencies as well. And we also showed that with this treatment, we never quite restored levels of monoinnervation in the MNTV. So without the return of microglia in animals that are maintained on this PLX 5622 CSF1R inhibitor, we really were not able to see restoration of these features of auditory system development. Okay, so this tells us that with these broad strokes that microglia have really profound effects on the maturation of the auditory system. And we've started to think about, well, how do they work? What are the molecular pathways that they use? And I showed you earlier that there are several signaling pathways that are used by microglia to communicate with neurons. And we've begun to look at several of these in my lab. One of the ones that we looked at is called CX3CR1, which is a receptor for CX3CL1, which is also called fractalkine. And this is a cytokine that's used for signaling between neurons and glia. So here we have CX3CR1 on the microglia, and it responds to the CX3CL1 that is released from neurons. And we've shown that there's expression of CX3CL1 in the developing brainstem that really correlates well with the timing of the appearance of microglia. Okay, so we started to test what are the effects of deletion of CX3CR1. So in these animals, unlike the previous studies, they have microglia, and the only difference is the microglia don't express CX3CR1. And just as a point of reference here, these animals also express GFP in driven by the CX3CR1 gene. So the microglia are shown in green. So we first looked at GFAP, this marker of mature astrocytes. And we found that unlike the previous study, there's actually a significant increase in GFAP levels when we looked at postnatal day 14. So it looks like astrocytes are maturing at a normal or slightly faster rate than in controls. We next looked at synaptic proteins, and what we found is that there's normally a decrease in V-glut with age, and we didn't see it with the mutation. And the same thing was true with VGAT. And one of the most interesting results here is that we found that another presynaptic inhibitory marker, GlyT2, was elevated at P14, but not at P8. And this is a marker that we know decreases with age, and it may coincide with elimination of inhibitory synapses on MNTB neurons. So this suggests that the CX3CR1 mutation is important for the pruning of inhibitory neurons on MNTB. To look at this a little bit further, we did the same sort of engulfment assay that I showed you earlier. We stained for GliaT2 in heterozygote CX3CR1 neurons, which have a fairly normal development, but all the microglia are labeled in green. And you can see that there is GliaT2 co-localized within these microglia. And again, we saw that there was a significant amount of co-localization with this lysosomal marker, CD68, and you can see some of those here. So this suggests that, in fact, microglia are engulfing inhibitory presynaptic terminals during development. And it's possible that in the CX3CR1 mutation, this process is impaired. So that suggests that microglia prune inhibitory synapses in a CX3CR1 dependent manner. So we then looked at pruning using the same technique, and we found that there was absolutely no difference in the levels of monoinnervation in MNTB neurons. And we also looked at the distribution of the polyinnervated neurons. There was really no difference whatsoever. Okay. So one of the interesting things that we looked at here is we know that there are several gradients in MNTB, many gradients on many molecules. And one of the things that we know is that there's a gradient of cell body size that we normally see. And so this is the gradient of cell body size is not there in the CX3CR1 mutants. So they seem to have slightly larger cell size, particularly in the medial region. So we looked at that, and that was fairly consistent. So we also found that there is actually a gradient of calyx size. I didn't talk too much about the sizes of calyces, but we did see that there's a gradient of calyx size, and that gradient seems to be missing in the CX3CR1 mice, and that's probably accounted for by the larger calyces that we saw in the medial region. Okay, and then finally, we looked at what happens in the ABR in the CX3CR1 animals. And this is something that's been studied this has been looked at before, but not really quite at this level. We didn't see any difference in click thresholds as previously determined, but we saw that they have a slightly lower tone thresholds than wild type mice. And when we looked at the latencies, which were the interesting phenotype when we eliminated microglia, we found that ABR latencies were actually shorter in the CX3CR1 mice than in wild type controls. And this was actually really a surprising feature. So this suggests that the signaling pathway CX3CR1 is really not accounting for the defects that we see when we deplete microglia. Similarly, we showed that calyx pruning is not impaired, GFAP is not impaired. In fact, it's significantly elevated. And the ABR's latencies are shifted in the opposite direction. So, so this suggests that CX3CR1 really doesn't account for most of the differences that we saw with the microglia depletion. However, we did see a potential role for CX3CR1 that in regulating the pruning of inhibitory synapses. Okay, so in conclusion, we see that microglia have several important roles in the maturation of the auditory circuits. We saw that microglia affect astrocyte maturation, and this is really an interesting factor because we know that microglia are not only communicating with neurons, but they're also communicating with other non-neuronal cells, including astrocytes and oligodendrocytes. So microglia also, microglia depletion impairs pruning. We know that there is synaptic engulfment by microglia, both of excitatory and inhibitory synapses. And as I mentioned before, since microglia are communicating with astrocytes, there may be multiple mechanisms by which these synapses are pruned. And one of the reasons we wanted to investigate this is that while it's been shown that microglia are really good at pruning synapses in many parts of the brain, all of the synapses that have been studied have been small Bhutan-like synapses, whereas here we're looking at pruning of really large calyceal-like early inputs. And so we weren't sure if microglia were really big enough to participate in the pruning of these large synapses. So one thing that we see is that given the increase in threshold in the ABR, we think that there are both peripheral and central effects of BLZ treatment. And one of the things we'd like to do in the future is investigate how these different effects are related versus distinct. So we know that there are central effects of microglia depletion, but to what extent are these related to the changes that we see in what may be peripheral hearing? And finally, we started looking at CX3CR1, and it doesn't look like it's accounting for the large-scale effects that we see with microglia depletion. But we're still looking at some of the other signaling pathways, including C1q and C3. And we're currently doing studies to investigate whether these other signaling pathways look more aligned with what we saw when we depleted microglia. Okay, and I really am extremely grateful to two very talented scientists who worked on these projects, Gidri Milinkevičiūtė and Seema Chokar, a postdoc and graduate student in my lab, did the majority of the studies that I've told you about, along with Caden Henningfield, Emily Castro, and Minan Din. We also have several collaborators that are working on these projects with us. So with that, I will take any questions that you have, and thank you for your attention. Mark, I think you're muted.
Hey, yeah, thank you. That was great. Thanks. I will. We have time for questions, and I'll give the question askers the option to either raise their hands and I'll call on them. They can ask directly, or they can send questions via the chat and I will ask them for them. And to get things going, I'll start with one. I was really curious about the different effects of knocking out CX3CR1 on the pruning of the calyces versus the inhibitory neurons. And I wonder if you know anything about the expression of the ligand CX3CL1 on those two different neuronal populations, and perhaps, you know, whether inhibitory. Yeah, go ahead.
Yeah, that's a really great question. So we did see CX3CL1 in MNTV neurons and in the other principal neurons of the auditory brainstem. But we haven't looked at the expression in the inhibitory neurons per se. But one thing that's really interesting about this is that we already know from some studies that were done in the cerebral cortex that different populations of microglia prune excitatory versus inhibitory synapses. And they do so using different types of signaling pathways. So it could represent distinct mechanisms for these two types of synapses.
Thanks. And there's a related question here in the chat asking, why do neurons release CX3CL1 in general? And I assume that they're asking about the role of fractalkine signaling in mediating communication between neurons and microglia. Maybe some very basics about that signaling process.
Yeah, so there are lots of reasons why neurons have to communicate with microglia. Microglia were first really understood in the context of immunity and neuroinflammation. And this is one of the ways that neurons can tell microglia that they need help or that they need some kind of adjustment. The roles of microglia in development are relatively new. We're just now starting to learn about what they do during development and how they do it. We know that their patterns of gene expression are completely different during early development versus later, so that the kinds of signaling that happen between neurons and microglia will lead to completely different sorts of effects. So for instance, there's some evidence that CX3CL1 is a marker used to indicate which synapses are retained and which synapses need to be eliminated. There's also a lot of evidence about CX3CR1 and its potential communication with oligodendrocytes. So it's a really great question. Cytokines are really interesting because they provide this mechanism of signaling in multiple different contexts. And what the neurons are going to do with those contexts depends on their patterns of gene expression and what they're poised to do from a cellular molecular standpoint. I hope that answered the question.
Yeah. Question regarding the elimination of microglia, specifically wondering whether there are any clinical conditions that lead to depletion of microglia, and how inflammation affects microglial function. And I suppose, it might also be worth touching on the use of these CSF1R inhibitors in a clinical context.
Okay, so there is a clinical setting in which patients have a mutation in CSF1R, and a homozygous mutation is almost always lethal. And there's a syndrome also where there are heterozygotes, and it's rather uncommon, but there are some patients that have been reported that have that. The interesting thing about the CSF1R inhibitors is that they are actually in clinical trials now, but not for development. So the original idea that some of our colleagues had about this is that in neurodegenerative disease, sometimes it's thought that microglia are kind of old and sick, diseased microglia, and they're no longer able to do their sort of cleaning up functions. And the original idea was, well, what if we got rid of them and replaced them with new happy and healthy young microglia? And in many studies, it's shown that this actually works. So especially since the microglia repopulate, and this has been shown to improve outcomes in animal models of Alzheimer's disease, among other things, improves brain plasticity. And I know that there are some of my colleagues now are actually looking at creating purified human microglia that can be used in cellular therapy for neurodegenerative disease. And you can actually, you can use CRISPR to correct mutations in them and basically replace diseased or mutant or sick microglia with healthy ones. And so this is a very exciting potential area of investigation.
A question about material presented in the first part of the talk. You showed that the shape factor of calyces was unaffected by microglial depletion. Did the arborization proceed normally in the absence of microglia?
We didn't measure this quantitatively, but we did see that in both cases, they appeared to grow at the same to the same levels and show high levels of reticulated fenestration in their morphology, but we don't know about the we didn't measure it quantitatively.
Another question raised about the repopulation of microglia after you stop treatment with the CSF1R inhibitors. And the question here is, do you know where the new microglia are coming from? Is there a small population of precursors that remain, or is there any evidence for infiltration of new monocytes or macrophage-like cells that can then repopulate the brain with microglia?
So that's a really great question, and one that we're looking into at the moment. We're very interested in this idea that these repopulating microglia resemble developing microglia, that they basically, one hypothesis that we have is that these new microglia basically recapitulate the developmental program so that they can enable maturation of an auditory circuit and in age much later than we normally see these developmental functions going on. So the fact that the microglia emerge from lateral to medial is also really compelling. So we think maybe the new microglia are also coming from the same source as the original microglia, but that's something we just don't know. One project that we have in mind is to characterize the repopulating microglia using spatial transcriptomics to try to identify what cell types are contributing to these repopulated cells. And potentially fate mapping as well, so we can investigate this. We know from other studies of microglia repopulation that they can come from a variety of sources. So in the early studies that were done in the Green Lab, they showed that when they eliminated microglia with PLX, the repopulating microglia came from these sort of stem cell-like cells that were left behind. So they didn't eliminate 100% of the cells. There were a few left behind, and those could provide repopulation. But there also may be other types of cells that can help contribute, including macrophages, monocytes, and those are our best candidates at this time. And so, yeah, that's an excellent question, and one that we're really excited about studying. And one of the really interesting things about this is that, you know, we know that microglia in the brainstem do different things compared to microglia in other parts of the brain. This is one of the very interesting things about them, that we know that their gene expression patterns are highly, highly diverse, especially during development. So microglia in one part of the brain are sort of specialized to do what that part of the brain needs it to do. And if new repopulating microglia can take on these roles, that means that the instructions for specifying the functions of the microglia remain within the brainstem nuclei themselves.
Yeah, I know from my own reading that most people who study this, tend to focus on the cortex and hippocampus who study microglial depletion and repopulation. But I was wondering, do you know if there's any evidence for a small pool of precursors on the lateral side that might account for, this, lateral-based repopulation?
We don't have, we don't know, but it is definitely something that we've been looking into in one of our planned future studies.
Great. Another question, as you mentioned, progenitor cells can convert into microglia. And they're wondering during hemorrhage or exposure of blood cells to the MNTB, whether conditions like that might affect neuronal pruning. I mean, I'm assuming here that they're asking, in situations where there's been injury and other inflammatory cells have been allowed to enter the brainstem.
I think that's a really great question, and one that's especially important when we think about traumatic brain injury or stroke, or other kinds, anything that's going to breach the blood-brain barrier. And I'm not sure how that specifically would affect neuronal pruning, but we do know that that neuronal pruning is a feature of lots of disease states. So abnormal pruning or maladaptive pruning happens. And so while we have normal pruning going on mediated by microglia in early development, there may in fact be pathological pruning that happens in any one of these situations. And we have not looked specifically at how that might influence development, but it's highly likely to have important consequences in disease or injury.
Okay, well, it's exactly 1 hour. I think this would be a good time to wrap things up. I'm sure if anybody has any remaining questions, they can email Dr. Cramer directly, and she'd be happy to answer them. And I'd like to thank Dr. Cramer for a great presentation, and also thank all the attendees for showing up, and look forward to seeing everybody again virtually in the next one of these ARO seminars. Thanks again.
Thank you.